Hello everyone, welcome to our YouTube channel. My name is Dr. Chernik Volodymyr. Today we are diving again into a fascinating topic in the world of advanced medical science: xenogenic, organ-specific and brain-specific precursor stem cell xenotransplants. Let's explore some of the most frequently asked questions about these innovative cells and discover their potential to revolutionize medicine. We've already made a series of videos on that topic, so please don't forget to check them out. Don't forget to hit the subscribe button if you enjoy the content. And from here we start.
What role do xenogenic stem cells play in the field of regenerative medicine?
In regenerative medicine, targeted organ-specific precursor stem cell xenotransplants offer a potentially unlimited supply of cells for therapies to repair or replace damaged tissues and organs. They are particularly valuable in scenarios where human cells are not suitable or probably sufficient, thus expanding the tools available to regenerate tissues, alleviate symptoms, and cure underlying conditions.
What conditions could potentially be treated with xenogenic stem cells?
Targeted organ-specific and brain-specific precursor stem cell xenotransplants hold promise for treating a variety of conditions, particularly where human donor cells are scarce or where autologous cells, means from the patient, are not viable. Some promising areas include, for example, diabetes, by differentiating into pancreatic islet cells that produce insulin, potentially offering a functional cure or significant treatment improvement. Then next, heart disease. So repairing damaged heart tissue after myocardial infarctions by differentiating into cardiac muscle cell improves heart function as well. Then talking about liver diseases. Liver cirrhosis might be treated by cells that can repopulate and restore liver function. Then neurodegenerative diseases, it's like a golden standard in stem cell treatment. So, such as Parkinson's disease and Alzheimer's, where they could differentiate into neuronal or glial cells to replace damaged or dysfunctional brain tissue. In Parkinson's disease, the transplantation of dopaminergic neurons derived from xenogenic stem cells could replenish the depleted dopamine levels. Then talking about spinal cord injuries, where the stem cells could aid in repairing or regenerating nerve pathways, potentially restoring some levels of function and mobility. And then I should say that actually this list is potentially never ending. Using targeted organ-specific cells creates an excellent potential for professionals to treat almost every disease, whether congenital or chronic, depending on the patient's medical background and, of course, possible contraindications. Because of ongoing research, we're expanding the horizons of knowledge, and as science advances, we find more and more strategies and modalities to treat various diseases and disorders.
How do the precursor stem cells integrate into human tissues?
When transplanted into human tissues, organ-specific precursor stem cell xenotransplants undergo a complex series of events to integrate and function effectively. The primary steps include, first of all, talking about the targeted transplantation, I'd like to remind that progenitor precursor stem cells from a specific organ like brain or pancreas, for example, are more likely to successfully colonize and regenerate the same organ in the host body. This is due to their inherent programming, which includes specific adhesion molecules, receptors, and response to local growth factors. It is all related to the principle of organospecificity and will finally lead to more effective tissue repair and regeneration. So, these cells need to locate or home to and attach or engraft to the appropriate tissue. Homing of the transplanted precursor cells to specific organs is a multi-step process that includes cell migration, into the blood vessels and entry into the bloodstream, attachment and rolling in the vessel lumen, adhesion and extravasation across the vascular endothelium, and migration through the tissue stroma. The migratory ability of the cells is possible due to the biochemical signals from the target area, reception system, and chemotactic properties of cells. Fetal precursor stem cells have a very high potential for migration and homing to the sites of injury. This process involves the migration of cells into the bloodstream. We say that's probably happening by rolling. Transendothelial migration through the vascular walls and invasion into the target tissue. The expression of homing receptors that match ligands in the target human tissue facilitates cells' migration to and retention at the site of injury or disease. Usually homing takes a few days, sometimes up to 72 hours. Retention at the site of the injury or inflammation won't be possible without a principle of homology as well. And that is something crucial for us for understanding the entire process of precursor progenitor stem cells implantation. Homology refers to the degree of similarity in genetic sequence, structure, and embryonic origin between organs or genes from different species. In the context of xenogenic stem cell transplantation, this principle is crucial because it suggests that organs or cells that are evolutionarily conserved and equal may function similarly across species. And that is why stem cells are capable to integrate and function properly if the target tissue in the host species shares significant evolutionary and structural similarities with the tissue from the donor species. At the same time, most of the cellular functions and regenerative processes are conserved across species, which makes xenogenic stem cells effective in a foreign biological environment. Next, once in place, precursor stem cells are programmed to undergo a specified process of specialization into a single cell lineage exclusively. This process is directed by the local microenvironment in the tissue. Finally, the new cells must integrate into the existing tissue network, forming connections and performing the required functions, whether insulin production in the pancreatic beta cells or neurotransmitter release in neurons, directly through the regeneration and remodeling of the corresponding organ or through the paracrine effect.
What is the paracrine effect? What is the mechanism behind it?
The paracrine effect in stem cell biology refers to the mechanism by which stem cells exert their therapeutic effects indirectly through the secretion of soluble factors, rather than by directly replacing damaged cells through the engraftment. This concept has become increasingly significant as researchers understand how stem cells contribute to tissue repair and, of course, regeneration. So, talking about the mechanism involved, we should say that stem cells produce and secrete various bioactive molecules such as growth factors, cytokines, chemokines, and extracellular vesicles, including exosomes. These substances can influence the behavior of neighboring cells and the surrounding tissue environment. The role of secreted factors is massive and dramatic. So, growth factors promote cell growth, proliferation, and differentiation. For example, fibroblast growth factors, or FGF, and vascular endothelial growth factor, VEGF, help in the repair and regeneration of tissues by stimulating angiogenesis, which means formation of new blood vessels, and cellular proliferation. Cytokines and chemokines. These molecules mediate immune responses and attract immune cells to sites of injury, helping to modulate inflammation and facilitate healing at the same time. extracellular vesicles and exosomes. These are small, tiny vesicles released by cells that can carry proteins, lipids, and RNA. They play a critical role in cell-to-cell communication and can modulate recipient cell behavior, potentially even transferring genetic material. It all implicates an essential role in the paracrine effect for tissue repair and regeneration, modulation of the immune system, and anti-inflammatory effects. The paracrine effect underscores a shift in how we view stem cell therapy, from focusing on replacing damaged cells to enhancing repair mechanisms already present in the body. This understanding opens up new avenues for treatment and therapeutic innovation, expanding the potential applications of stem cells research far beyond direct cell replacement.
How does the public perceive the use of xenogeneic stem cells, and how can their acceptance be increased?
So until today, public perception has been mixed, influenced by factors such as cultural beliefs, understanding of science, and ethical viewpoints. And I would say that the strategies to increase acceptance include, first and foremost, education, providing clear, transparent information about the benefits, risks, and ethical consideration to demystify the technology and address misconceptions is so important. And it's a huge and vital part of our everyday lives as professionals in the field. Patient testimonials and success stories are essential. Sharing positive outcomes and ethical handling cases can humanize the technology, and for sure it can demonstrate its value. Until the day we have published dozens of publications and books. Please follow the link to find them in the description. Okay? So, regulatory insurance is next, and it is also a must. Demonstrating that robust regulatory frameworks are in place to ensure safety and ethical compliance can also help build trust. And I'd like to tell once again, in our practice we use specific pathogen-free animals, sheep or rabbits. Specific pathogen-free SPF animals refer to laboratory animals, sheep or rabbits, that are carefully bred and maintained in controlled environments to ensure they are free from viruses, any kind of bacteria, parasites, and other pathogens that might interfere with scientific research. Emphasizing once again on the fact that SPF programs typically involve rigorous testing and monitoring to ensure the animals' continued health and pathogen-free status. This commitment to quality insurance helps maintain the integrity of the stem cells derived from these animals.
What are the main routes of the cell transplantation?
There are two main routes of cell transplantation: directly to the target tissue or systemic. Direct implantation is possible either in superficially located areas like dermal cell grafts for skin defects, intraarticular injections or intrathecal administration, etc., or to the relatively accessible tissues and organs like ultrasound-guided implantation into perinephric space or liver, endovascular procedures via laparoscopic approach, etc. The systemic route of administration is either intravenous or intramuscular. The intravenous route of administration is feasible only for cell preparations with a rather low amount of administered cells because otherwise it would carry significant infusion-related risks like thrombosis or embolism. Another disadvantage of the intravenous route of administration is the tendency of the implanted cells to be trapped in the lungs, which results in an extremely low percentage of cells performing actual homing into the target tissues. Literature suggests that intramuscular injection is becoming more and more popular method of stem cell delivery, with promising results seen in the treatment of various diseases, over plenty diseases. In contrast to the shorter therapeutic life of mesenchymal cells delivered via intravenous injection, intramuscular injection of xenogenic stem cells could potentially treat both distant and systemic conditions with sustained benefit owing to the longer dwelling time of active secretory cells and has been clinically proven to be absolutely safe. Also, sometimes people ask me about the inhaler method of stem cell delivery, how good it is, what conditions we can treat using this delivery method, and so on and so on. And here I'd like to say, so long story short, this method is used primarily for respiratory diseases where inhalation delivers stem cells is an aerosol. It is an ideal method for pulmonary conditions as it delivers cells directly to the lungs and avoids the need for injections or surgical procedures. So cells reach the lung tissue quickly, which is beneficial for acute conditions such as acute respiratory distress syndrome, ARDS. Still, using the inhaler method is limited to treatments involving the respiratory system. Dosage control and deep lung delivery can be possibly challenging in this case. Ultimately, to sum up, all I want to say that the best method is often determined by the specific clinical context, the nature of the stem cells used, the intended target, and the condition being treated. Tailoring the delivery method to these factors can maximize the efficacy and safety of the treatment.
How can xenogenic stem cells can traverse the blood-brain barrier and perform their function in the brain? What are the methods to prove this ability?
Yes, targeted brain-specific precursor stem cells, xenotransplants, can cross the BBB. And this ability is part of what makes them valuable for treating neurological disorders. Following the completion of the implantation process, stem cells utilize the body's signaling mechanisms to accurately navigate towards particular locations, such as brain and different parts of the brain. So BBB itself is the highly selective barrier composed of endothelial cells with tight junctions, astrocytic end feet, and pericytes. For stem cells to cross the BBB, they must first adhere to the endothelial cells. This is mediated by cell adhesion molecules such as integrins and selectins. Following adhesion, stem cell can cross the BBB either via paracellular routes between the endothelial cells or transcellular routes through endothelial cells as well. It is also possible. In conditions where the BBB's permeability is increased, such as inflammation or injury due to certain degenerative conditions or trauma, stem cells find it easier to cross into the brain. The inflammatory milieu, characterized by the presence of cytokines and chemokines, enhances the expression of adhesion molecules on both stem cells and BBB endothelial cells, facilitating transmigration. Also, we know that some stem cells express MMPs, which are enzymes capable of degrading the extracellular matrix components around the BBB. This degradation can lead to increased permeability of the barrier, facilitating the transmigration of stem cells into different areas of the brain parenchyma. Also, the possibility of xenogenic stem cells crossing BBB and surviving was showed by a number of radioisotope studies. I'd like to demonstrate a few more. This research employed MRI, magnetic resonance imaging, and immunostaining to track the presence and integration of adipose-derived stem cells in the rat brain over an extended period of 6 months. The longevity and localization of the cells were primary focuses. In the next study, the researchers have shown that human neural progenitor cell xenografts survived extendedly in the brain of immunocompetent rats. The use of immunohistochemical analysis allowed researchers to observe the survival and integration of human neural precursor cells in the rat brain. Additionally, electron microscopy was utilized to examine these cellular interactions at the microscopic level. In the following study, researchers combined the administration of mannitol, which temporarily opens the BBB, with the injection of stem cells. They then used imaging and histological methods to track the migration and localization of these cells within the brain. And there are plenty of other studies that confirm the fantastic regenerative ability of brain-specific precursor stem cell xenotransplants. And another one thing that I want to emphasize on is that the methods that allow us to observe their activity are crucial for proving the ability of xenogenic organ-specific stem cells to cross the BBB and for assessing their survival and integration into the particular area of the brain tissue, thereby supporting their essential utility in therapeutic application for neurological disorders. Thank you so much, ladies and gentlemen. Wishing you great health and all the best, and I hope that this information was valuable and interesting for you. Thank you so much. See you soon.